Hi everyone, it's Manny from MedCoach TM, and today we're going to be talking about T cells. So T cells are a part of the adaptive immune system, and the adaptive immune system takes a little bit longer to activate than the innate immune system. It also has specificity and it has memory. So what this means is that only specific parts of the adaptive immune system will bind to specific antigens, and it also has memory. So if the body is hit with the same pathogen the second time, the adaptive immune system will have a stronger and faster response to this antigen. So it has memory. Now, T cells are important. Something to remember about T cells is, again, the specificity, right, guys? So T cells are going to react to antigen only if they are presented by the APC. What is the APC? The APC is the antigen presenting cell, and what the antigen presenting cell has is this MHC2 or MHC1 that will basically present the antigen to T cells, to the T cell receptor. So this is extremely important, because without this APC presenting this antigen, T cells will not be activated. They're not going to go and start doing what they're supposed to be doing. So this is usually the way that it works. So we have some sort of APC. So we have the APC, and we have this antigen that comes to the APC. The APC engulfs it, does whatever processing it needs to, and then we have an MHC molecule outside, which then allows for this this antigen to be presented. Then we have a T cell over here that has a T cell receptor, which is going to notice this and bind onto it. This is going to activate this T cell, and this is going to make the T cell very happy. So another thing that we want to talk about is the T cell receptor. So as mentioned earlier, we have a T cell, and this T cell has a T cell receptor on it. However, the problem with this T cell receptor is that it's too short. It doesn't have a good enough ability to properly send signals inside of the T cell. So this is when our friend CD3 comes in, and CD3 is our friend because what it's going to do is it's going to help the signal that's coming in, first of all, come in all the way inside, so that this T cell is going to be activated and be happy. So this is extremely important to remember, guys, this CD3 complex that surrounds the T cell, because a CD3 is what you can use to recognize whether or not there are T cells present. So for example, let's say you get a flow cytometry, right? So you have a flow cytometry, and let's say that you have some other antigen, maybe like CD20, so this would be a B cell, but then you have a CD3. And let's say that you get a question and it kind of looks something like that. So that's your flow cytometry. And your job is to figure out, by looking at this, what kind of cells are present. And maybe they might ask you, that this is the flow cytometry, what could possibly be going on right now with the person, with the patient? What's going on? Do they have some sort of immunodeficiency? Do they have an autoimmune disease? Who knows? But by looking at this, you need to determine what they have. So let's take a look. So from here, you can see that, first of all, we have a low amount of CD20, and we have a low amount of CD3. So this means that we have a low amount of B cells, but we also have a low amount of T cell. And by using this information, you can tell or answer the question and let them know what you think is going on with this patient. So that's something that's extremely important that you need to remember. I would say that this is an extremely high yield fact. I'm going to double highlight it, or actually I'm going to triple highlight it, and I'm even going to put a star beside this so that you remember that this is an extremely high yield fact. And just a small point to remember is that in regards to T cells, there are two subsets. There's CD4 helper T cells and CD8 cytotoxic T cells, which we're going to be talking about in the upcoming modules on what the differences are between those T cells.